Today we'll be looking at DNA replication, specifically in prokaryotic cells. In the top we have the leading strand, which is 5' to 3' and has continuous replication, while the lagging strand is 3' to 5' and is discontinuous replication, which means it will produce Okazaki fragments. DNA polymerase I removes primers and fills in gaps with a 5' to 3' exonuclease activity, while it proofreads with a 3' to 5' exonuclease activity. DNA polymerase III is the enzyme mainly responsible for DNA synthesis, and it proofreads with a 3' to 5' exonuclease activity. Helicase breaks the hydrogen bonds and separates the strands to allow replication to occur. Single-stranded binding protein stabilizes the strands so they don't reconnect. Gyrase is a topoisomerase, so it prevents supercoiling from forming, and it's located behind the replication fork. Primase lays down the RNA primers, and DNA ligase seals the gaps between the Okazaki fragments with phosphodiester bonds. Now let's look at this entire process in action. Helicase is going to break the hydrogen bonds connecting the top lagging strand to the bottom leading strand, and gyrasing prevents supercoiling. Binding proteins are going to attach to prevent these strands from reconnecting. Primase is going to leave RNA primers on each of the strands and multiple on the lagging strand because DNA polymerase III reads from 3' to 5'. Starting near the origin of replication, DNA polymerase III connects to the primer and does a single continuous DNA synthesis from 3' to 5'. On the lagging strand, DNA polymerase III has to jump from each RNA primer, still going from 3' to 5', creating these Okazaki fragments. And now we have new direction on our new DNA. Now DNA polymerase I reads from a 5' to 3' and has exonuclease activity. So it's going to remove these primers and fill in the gaps with new DNA. If we look closely at the new DNA, we can see gaps are formed between the new DNA created by DNA polymerase I and the Okazaki fragments created by DNA polymerase III. And so DNA ligase is going to come in and fill these gaps with phosphodiester bonds to create a continuous strand of DNA. So what would happen if DNA polymerase I didn't have its 5' to 3' exonuclease activity, which would remove primers on each of the new strands of DNA? So, if this was going to happen, DNA polymerase would attach to the primers, but unable to remove them to fill with new DNA. And thus, the new DNA will be non-viable. They'll have RNA still a part of the strand. And so, the next cell that would receive these DNA strands would ultimately die because there's RNA a part of it.